Hi everyone, am I audible? Hi everyone, am I audible to you all? Hello. Hi Shrutik, hi everyone. How are you? Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and I think today you people are very happy because now you have the clarity on your syllabus. You know now your syllabus is reduced. So how are you people feeling? Tell me. How are you people feeling? And I'm sorry to start the class late. I think there is some confusion. I was live but on some another session, right? So I just checked it right now. Acha, you people are feeling very bad. Why? Why? What happened? What happened? What happened? I thought you guys are happy that now your syllabus is reduced. I am not happy, honestly. But that's my personal opinion. That's my personal opinion. Which bad news? Those chapters were easy, scoring your digestion and absorption, transport in plants. But not from this perspective that you can score better in that particular chapters. What I believe that if you don't know the basics, right? If you don't know what exactly is the active and the passive transport, what exactly is that facilitated transport, right? How the water is moving up. How can you understand photosynthesis? Right? In your batch, still, we taught that part. Right, that this is what transport in plants, that is what digestion and absorption, but still, if you don't know these basics, then how can you understand other systems? This is what I believe. So that's my perspective. So it's okay. Now your syllabus is reduced. Still, you will be having the options. Okay, so there are chances that you can score better. No need to get upset. So whatever you have now, the same time is there, but the number of chapters are reduced. See, initially you used to have 38 chapters in biology. Now you have 32 chapters in biology, but the time is same. So I think you can utilize this thing in a better way. And today around 2, right? Stay tuned to our channel. I will share the revised strategy to score 360 out of 360 in biology. We will talk about the reduced syllabus. Okay, we will talk about the reduced syllabus, and we will discuss that how can we manage this time to score better. Fine. And if cockroach is deleted and the frog is there, so it's okay, fine, we'll figure out. We will discuss that too. Don't worry about that. Don't worry about that. Okay, no need to get worried. But I think one thing is for sure, one thing is crystal clear there, that your molecular basis of inheritance chapter is still there, isn't it? Molecular basis of inheritance, it's still there in your syllabus. And now, because other 6 chapters are deleted, yes, there are chances that you will get more questions from physiology, you will get more questions from genetics. Okay, yeah, now you have to go for the new NCERT. You have to go for the new NCERT. I don't think that paper will be tough, to be very honest, Satyam. I think the level will be same. But yeah, now if you will check your previous AIMS papers, so you will see that actually the questions were not just on the memory basis, right? They were not just memory based questions, but conceptual questions were also there, right? Critical thinking kind of questions were also there. Are you getting it? So that's better. That's better. You think you think to answer that questions. It's not like that if your memory is good, then you're scoring good. No. If you can understand that concept, then you're scoring good. So that's a wonderful thing. And now, yes, today I will keep one session, students. I will keep it live so that I can answer all your questions. So I will keep strategy session today only, right? I will take that session. Don't you people worry about that. Fine. I know there are a lot of questions in your head. I know you all are baffled. You don't know how to proceed now, how to start now. But trust me, I will keep the session for that. Okay, I am going to keep the session for that. So happy. So one thing is for sure, that genetics is going to come. And yes. If you are expecting 44 to 50 marks in genetics, right? So now you can just raise that bar. Fine. Yup, it's reduced, Nandini. We'll discuss everything, Nandini. We'll discuss everything. Don't you people worry. So now I think what we should do is, right, we should continue this chapter. And today we are going to start transcription. Isn't it? That's what we are going to start. That's what we are going to start. So let's start the transcription, guys. Let's start the transcription. Let's not waste the time. Now we know that. Right now we have all the clarities. Now you guys cannot crib. You cannot say that, oh, we don't know what to follow, what not to follow. Now you know everything. Right? Now you know everything. So let's start the class, everyone. Just hit the like button and let's continue the topic. And let's start with the transcription. Okay? Which topic are we going to start today? We are going to start the transcription. And trust me, I will tell you each and every possible question from this chapter. Okay, done. So let's start from a very basic thing, and I know you all are well aware of it, and it's a repeated question in the NEET examination Sabha. It's a repeated question. Okay, so you know that when it comes to a cell is having the DNA, storehouse of all the information, right? The DNA can replicate, we know that. Right, replication will occur. So that DNA will form the mRNA, and that mRNA is going to form what? That mRNA is going to form the protein. That's what we know, isn't it? That's what we know. DNA to mRNA, mRNA to protein. So, bacha, when it is DNA to mRNA, this process is known as transcription. And I will tell you why it is important. When it is mRNA to protein, this process is known as translation. Now, see. Because DNA is the storehouse of information. DNA is having nitrogenous bases. DNA is having nucleotides. Right, bache? Right. Let's recall that portion. Let's recall that portion. So when it is your DNA, right? When it is your DNA, you know that it's a polynucleotide chain. There are so many nucleotides, right? It's a polynucleotide chain. There are so many nucleotides. There are nitrogenous bases there, sugars there, phosphates there, right? They have that information. Okay, they know what to form, what not to form. But your DNA cannot go to each and every part of your cell. Let's take the example. This is your prokaryotic cell having DNA in the cytoplasm. This is your eukaryotic cell having DNA in the nucleus. Right, having DNA in the nucleus, isn't it? Isn't it? So DNA cannot just go out. DNA cannot go to the mitochondria. DNA cannot go to the Golgi apparatus. Ki, hey, bro, do this, do that. Of course not. Of course not. DNA is the storehouse of information. So DNA will be there in the nucleus, right, in the eukaryotic cell, and it will guide other organelles, right? It is responsible for that metabolism. It is responsible for that enzymes, isn't it? It is responsible for that enzymes. It is responsible for everything. Are you getting my point, students? So here DNA, it will form a transcript of its information, right? Like let's say you have a book, you cannot buy that book. What will you do if you need that content? You will take the photocopy, isn't it? What will you do? You will take the photocopy, isn't it? Photocopy of that information, and then you can use it. Same is the case here. mRNA is just like the photocopy of the information present on our DNA. Okay, so that is the transcription. Now, very easy thing. In the case of prokaryotic cell, because there is no nucleus, so of course even the transcription will be there in cytoplasm. Even the transcription will be there in cytoplasm. And in the case of eukaryotes, because there is a nucleus, so the transcription will be there in the nucleus. That's what you need to remember. So, site of transcription. Site of transcription. In prokaryotes, that's cytoplasm. In eukaryotes, that's nucleus. Okay, that's nucleus. That's what you need to keep in your mind. And when it comes to the translation, your mRNA, the transcript of information, right, it is going to form the protein. Fine, bache, it is going to form the protein. So translation, in both the cases, translation, the site is cytoplasm. Where will that translation take place? It will be there in the cytoplasm, right? It will be there in the cytoplasm. So these are some basic points, but yeah, important they are. Okay, important they are. Done, bache? Okay. So that's what you need to keep in your mind. Sure, that's what you need to keep in your mind. Is that clear? Is that clear? So this is the basic process. And here, you know that when it comes to the DNA, gene, it is the gene which is the hereditary material. Sorry, it is the gene which is the unit of inheritance. Yes or no, Nandini? It is the gene which is the unit of inheritance. So even here in this topic, right, after this topic, we will also talk about gene expression. We will also discuss what exactly is the gene expression, how a gene expresses itself. So it's very simple. Gene is going to form the protein, students, right? Gene is going to form the protein. That's how a gene expresses itself. Okay, so understand the basics here. Understand the basics here. Is that clear? Is that clear, everyone? And yes, today I will take one session. I can see that message, Bache, Satyam. I will take one session. We will talk about the syllabus of the biology. I know there are so many confusions, right? So we will talk about the syllabus of the biology, and I will tell you a plan that how can you start, how you should proceed, right? You should make fresh notes or not. We will discuss each and everything. Okay? So just chill. I know it's difficult for you people to focus here because you're just thinking about your syllabus, but it is not going to help you out if you will just keep on thinking about it. Ultimately, what you have to do, actions, they matter the most, isn't it? Isn't it? So what do you have to do? You have to study. At least this chapter, we know this chapter is there in your syllabus, right? This chapter is a part of your syllabus. So let's do it now. And I will teach you this chapter today and tomorrow also the last part of this chapter, and then this chapter will be over. Okay, and now from coming week I'm going to take the one shots. Fine, I'm literally going to take the one shots. Okay, so focus here. This part clear? Yes, everyone, this part clear? So mRNA, ultimately mRNA is going to form from the DNA. So what do you want me to start first? The details of the RNAs, of the different type of RNAs, or the transcription? You tell me. You decide, and accordingly I'll proceed. What do you want me to do first? The detail of RNA or the transcription? Whatever you will say will go accordingly. What do you want first? The details of the RNA or the transcription first? Details of the RNA or the transcription first? About RNA? Fine. Let's go. So RNA is your ribonucleic acid. That's your RNA. And when we talk about the RNA, we know this thing, right? But we know this thing. It is also made up of many nucleotides. It is also having polynucleotide chain, right? It is also the polynucleotide chain. So when you talk about this RNA, I already told you about the RNA word concept. Let me mention it here. Okay, I already told you about the RNA word concept, that scientists, they believe that it is the RNA that came first. Okay, why? Because this RNA can also act like what? Like the nucleic acid. Moreover, it is also behaving like an enzyme, isn't it? Right? Right, bache? Right, bache? It can also act like an enzyme, and it is also acting like the nucleic acid. So we know it very well, isn't it? So when you talk about the RNA, you discuss two type of RNAs, right? Majorly, what are you going to discuss? We are going to discuss two type of RNA. One is your genetic RNA, also known as genomic RNA. One is your genetic RNA, also known as genomic RNA, and another one is your non-genetic RNA. Non-genetic RNA, right, bache? So these are the two things. So when it is the genetic RNA, means the RNA that acts like the genetic material. You know that here we are talking about the RNA that acts like the genetic material. Okay, how is it acting? It is acting like what? Like a genetic material. You can take the example of the retrovirus, isn't it? The virus in which it is the RNA which is the genetic material. We have many examples. Don't you remember TMV? Tobacco mosaic virus. Tobacco mosaic virus. Yes. Yes. And then you know about the HIV as well. That is also a retrovirus where RNA is your right? Where RNA is the genetic material. And moreover, you even know about your QB bacteriophage. That's a virus. And here also, the RNA is the genetic material. Are you getting my point? Are you getting my point? So this is what we know. Okay, so such type of RNA is your genetic RNA. Now when it comes to the non-genetic RNA, so you have three things here, bache. You have ribosomal RNA, you have messenger RNA, and then you have tRNA, that is your transfer RNA. So this RNA is not the genetic material, but it is helping in other duties. Like I just told you that mRNA is the transcript of the information present on DNA. It's acting like a photocopy of information. RNA, that is your ribosomal RNA, it is helping in the synthesis of the ribosome. tRNA, transfer RNA, it will help in protein synthesis. It will bring all that amino acids from the cytoplasm for what? For the protein synthesis. Okay, for what? For the protein synthesis. So firstly, let's start with what? Let's start with what? Let's start with the ribosomal RNA. Okay, let's start with what? Let's start with the ribosomal RNA. Okay, R here is ribosomal. Do you know that it is the RNA, right, which is forming your ribosome? Do you know that it is the RNA which is forming your ribosome? Do you know that? And this RNA is near about 80% of total RNA of the cell. Do you know about it? It is making 80% of total RNA of the cell. Yes, ribosomal RNA. Okay, ribosomal RNA. And now, bache, if you remember that when you talk about the eukaryotic cell, there is a nucleus, and here you are also having the nucleolus. Do you remember about the nucleolus? Yes. So this nucleolus is actually this nucleolus is actually known as ribosomal factory. Do you know that? Like ribosomes, it is the cell organelle which is helpful for the protein synthesis. It is the ribosomal factory. Yeah. It is the ribosome is known as the protein factory. Na? It is the one which will help in protein synthesis. Same way, when you are talking about the nucleolus, it is considered as a ribosomal factory. Why? Because this nucleolus is going to form your ribosomal RNA. It is going to form that ribosomal nucleoproteins. Right, so this nucleolus is going to form the things which are required for the formation of ribosome. That's why we used to call it as ribosomal factory. Clear, bache? That's why we used to call it as ribosomal factory. Is that clear? Is that clear? 80% of the total RNA of cell is your ribosomal RNA. And who's going to form this rRNA? This is your nucleolus. What is it? It is your nucleolus. Is that clear? Now, bache, it is the most stable type of RNA. Do you know that? Your ribosome RNA. Most stable type of RNA it is. Now you know that when you talk about a prokaryotic cell, that is your bacteria. Okay, that is your bacteria. Here you will get 70S type of ribosome. And in the case of eukaryotic cell, I hope you know that there will be 80S also. There will be 70S type of ribosome also. Do you know about it? When you are talking about the prokaryotic cell, you have 70S type of ribosome. What is this S here, bache? It is the sedimentation coefficient. It is the Swedberg unit, right? It is the Swedberg unit. It is the sedimentation coefficient. Okay, and here in the eukaryotes, 80S ribosome is present in cytoplasm and 70S ribosome is present in organelles. Which organelles? It is your mitochondria and the plastid. Isn't it? Isn't it? This is what you know. Don't worry, I will relate it with ribosomal RNA. Just wait for some time. VK bache, you are saying, bache, 80% of the total RNA means it's quantity, it's amount. It's amount, VK, it's quantity. Okay, so now when it is the 80S ribosome, it is having 60S. It is made up of two subunits, large and small, right? And even the 70S, it is also made up of two subunits, large and the small. Clear bache? Large and the small. So these larger unit and the smaller unit, they are made up of different types of ribosomal RNAs. Okay, they are made up of what? They are made up of different types of ribosomal RNA. For an example, this one is having 5S ribosomal RNA, 23S, 23S ribosomal RNA. Your 13S is made up of 30S is made up of 16S ribosomal RNA. Right? So that's how you name it. Okay, that's how you name it. That's why I'm relating it here. Okay, so ribosomal RNA is the one which will form the ribosome. Ribosome is very important for protein synthesis, right? So nucleolus in the eukaryotic cell is going to form ribosomal RNA. So when you talk about the ribosome, 70S and the 80S is there. 70S is having two subunits. Larger is 50S, smaller is 30S. Larger is having these ribosomal RNA. Smaller is having this ribosomal RNA. Okay, and same is for this 80S. Okay, the same is for this 80S. Do you know about the 80S? Yes, do you know about the 80S? So here larger one is having 5S ribosomal RNA. It is having 5.8S ribosomal RNA. It is having 28S ribosomal RNA. And when it is the 40S, it is made up of 18S ribosomal RNA. That's why I'm telling you this. This information is important for your NEET. Okay. This information is important for your NEET. And it is not just the ribosomal RNA which is making the ribosomes, it is also the ribosomal proteins. Clear, bache? It is also the ribosomal protein. So this part clear. If there is any doubt, do let me know. Yes, do let me know. Done? Done. Sure? Okay, so because this ribosomal RNA is going to make the ribosome, so you can say that it is providing the sites for the attachment of the tRNA, for the attachment of the mRNA. So it is helping in the protein synthesis. Fine, bache? It is helping in the protein synthesis. So next is your mRNA. Usually when I ask to students now, according to them, it is the mRNA which is having maximum amount in a cell. Right? This is the most common mistake, bache. Literally, it is the most common mistake. Most of the time, students used to say that the quantity of mRNA is more than that of other RNAs. But it is not like that. It is your ribosomal RNA which is making 80% of the cell. And then when it comes to the mRNA, it is near about 5% of the cell. Fine. It is near about 5% of the cell. And when it is the tRNA, that is your transfer RNA, then bache, that is your 10 to 15% of the cell. That is what? That is your 10 to 15% of the cell. So this mRNA, it is making 1 to 5% of total RNA of cell. First point. Secondly, it is the least stable RNA. Fine, it's the least stable RNA. And now in the transcription, we'll be discussing about this mRNA now. So it will also be synthesized in 5' to 3' direction. It will be having that nitrogenous bases. Actually, it is having the information for the synthesis of the protein. So that's how it is important. And that's why we call it as messenger, because it is having that message to form that particular protein which is required. Is that clear? Is that clear? Next is bache your tRNA, and tRNA is very important. Okay, next is what? Next is your tRNA, and the tRNA is very important. So T stands for transfer RNA. T stands for what? T stands for transfer RNA. Clear, Shrutik? What is this T? T stands for transfer RNA. Clear? Clear. It is also known as adapter RNA. It is also known as soluble RNA. Okay. It is tRNA. So it is very important for the protein synthesis. So when you talk about tRNA, it's two-dimensional structure. Right? That's important, bache. Question can come from this part. So when it comes to it, when you talk about its two-dimensional structure, it's clover leaf like. Okay, what type of structure it is? Clover leaf like. And when you talk about its three-dimensional structure, right? That is your 3D structure. It is inverted L-shaped, right? It is inverted L-shaped. So that's important for NEET. Fine, that is important for NEET. Clear, bache? Sure, soluble RNA. And this tRNA is making 10 to 15% of RNA of a cell. Clear, bache? It is making 10 to 15% of the total amount of RNA of a cell. Done? Done. Is that clear? Its structure is very important. Its structure is very, very, very important. Fine. Fine, fine, fine. And one more thing that you need to remember here, it is the smallest RNA node. It is the smallest RNA node. Is that clear? Okay. So now we need to discuss about its structure, and that's important. Okay. The structure of tRNA is literally important. Okay. Wait, I have added the diagram for you. So here you guys can see, although it's related to protein synthesis, but still it will give you idea about the structure. This is your clover leaf like, and this one will be the inverted L. I'm looking for another pictures if I'll get it. Nope, I don't have that. So here you people can see. Clear, bache? This is your tRNA. This is your tRNA. This is inverted L shaped. Okay, this is three-dimensional structure, and that one is your clover leaf like. Fine, it was given by Holland. Done, bache? It was given by Holland. Okay? Okay? Yes. So I told you, no, bache? Least stable is your mRNA. Least stable is your mRNA. I just told you. So now let's understand the structure of your tRNA. Okay. These can you see this? Right? So it's 5' end. This one is your 3' end. This is your DHU loop. Okay, and this one is your T-Psi-C loop. This is the most important loop, that is your anti-codon loop, and this one is amino acid acceptor arm. Right? What is it? It is amino acid acceptor arm. We will discuss it. See, when I'm using the word DHU arm, okay, I'm talking about this portion, and of course it is also having the bases. Okay, okay, here basically some bases are there, these nitrogenous bases are there. Okay, so when I'm saying DHU arm, I'm talking about this particular portion, and when I'm saying loop, this complete structure is the loop. Okay, this complete structure is the loop like this. It is a loop. Okay, so here you guys can see this is the 5' end, this is the 3' end. But that's what you need to remember. Fine, that's what you need to remember. Right here, when it comes to the 3' end, here you are going to find a sequence. Okay, it's CCA. It's CCA. It's CCA. Yes, bache, what is it? It's CCA. Wait. CCA. Always, this is what you are going to find here at 3' end. Right? Literally, this is what you are going to find here at 3' end, that is C, C and A. Now, some students, they must be thinking that, ma'am, why are you calling it as CCA? We can also call it ACC. Isn't it? Isn't it, Subhashini? We can also call it ACC. Why CCA? isn't it? Why CCA, ma'am? Right? See, it's very simple, students. It's very simple. Whenever you talk about any, whenever you talk about any of the mRNA, okay, any of the RNA, be it it's mRNA or any other RNA, when you read that sequence, you read it in 5' to 3' direction. That's what you need to keep in your mind, right? How are you going to read it? You're going to read it from 5' to 3' direction, 5' to 3' direction. So if I talk about that logic, so 5' to, if I have to read it in 5' to 3' direction, so it's going to be CCA. It's going to be CCA. What is it? CCA. And just to remember that, you can just simply remember that here there is a kind of sequence ACC. It is. That's all. But that's important. That's important. So in the paper also they can ask you, right? In the paper also they can ask you that where will you find the CCA, at 5' end or at the 3' end? It is your 3' end. And mostly when it is the 5' end, here you find guanine as the base. Okay, you are going to find guanine as the base. This is what they have observed. Fine, this is what they have observed. So this is your DHU loop, right, having 8 to 12 bases. Okay, 8 to 12 bases, right? Here you will find near about 7 bases. So this is the anticodon loop. So this is your tRNA, right, bache? This is your tRNA, and we also call it as adapter molecule. Let me tell you why. Now, I hope you know about the charges, right? We use that adapter, na? Adapter. We are going to plug it. We are going to plug it in, and then we have a space where we can put the wire as well, that cable as well, isn't it? Isn't it? Yes or no? Isn't it? Tell me quickly. Yes or no? Tell me. Tell me quickly. Isn't it when you talk about the adapter? It's like that. So when you look at the tRNA structure, if you will see it's different arms. So they are also meant for, joining to the different, they will also join to different parts, right? Imagine the structure is like this, right? This structure is like this. So this portion will fit in to your ribosome. This part will fit in to the enzymes. Okay. This part will fit into mRNA, and this part will fit in with the amino acid. So that's why we call it as adapter. Okay, that's why we call it as adapter. So it is having different arms to which mRNA, ribosome, enzyme, and even the amino acid can get attached. Okay, right? Even the amino acid can get attached. So that's what we are going to talk about. Fine. Okay, so see, amino acid acceptor arm, that is written here. Now, you know one thing, that when it is the 3' end, it will be having free OH group. Am I right or wrong? If it is the 3' end, it is going to have free OH group. Yes or no, Subhashini, Sabha? Yes? Yes. If it is the 3' end, definitely it is going to have free OH group. It is going to have free OH group. Yes or no? Yes or no? So now if it is having the free OH group, okay, so it is available for the bond formation with the amino acid. It is available for the bond formation with the amino acid. Now, when you talk about the amino acid, this is the basic structure of the amino acid. Am I right? This is a basic structure of any of the amino acid. Yes or no? Yes or no? This is the basic structure of any of the amino acid. Yes or no? You tell me. I hope you all know about it. I hope you all know about it. Yes. So this OH group is available for the bond formation with the amino acid. See, bache, I am saying that this arm is amino acid acceptor arm. So in the cytoplasm, there are amino acid. The job of tRNA is to pick up that amino acid and to take it for the protein synthesis. Okay. So it should have something where it can join that particular amino acid. So here you guys can see this OH group is available for the bond formation with amino acid. So when you look at the when you look at the basic structure of amino acid, so it is the basic structure of amino acid. It is the basic structure of amino acid, right? So it is having NH2 group, it is having C double bond OH group, it is having hydrogen, it is having R as well. Okay, so this COOH group of amino acid is available for the bond formation here. So basically the COOH of the amino acid, right? The COOH of the amino acid is going to form bond with this OH, and that's how your amino acid will get attached to this 3' end. That's why it is the amino acid acceptor arm. Telugu Ammai, yes, bache, syllabus is reduced. Today I will take a session dedicatedly for the deleted syllabus of biology. I will tell you the strategy. Please have patience, okay? Okay, so that's how the amino acyl bond is formed here, right? The bond will form here. Are you getting my point? A bond is going to form here. Clear? Clear? This is amino acid arm. So just write opposite. Okay, just opposite to this amino acid acceptor arm. This is the anticodon loop. But that's the part which will join with your mRNA. Anticodon. It is having, see, your mRNA will have code and your tRNA will have the anticode, right? Where it is going to join. Let's say if this is your mRNA, it is having AUG, right? It is having AUG. So complementary to it, the anticodon is going to be there on this anticodon loop. Are you getting it? It is going to be there on this anticodon loop. Like it can be UAC. Fine, it can be UAC. Are you getting my point? Are you getting my point? Yes. So that's how it is present. So here you will get what? You are going to get the anticodon. Fine, you are going to get the anticodon. Okay, I'll be more specific with the directions as well. So see, like here, it's UGC is written like this. Wait, I will tell you why. Okay, okay. Because this, here you will get AUG. This is the 5' end, this is the 3' end. So here you are going to get the another codon, UAC. Here you will get it. So that's how you have to keep it in your mind. Fine, fine, bache. So now these two arms clear. Amino acid acceptor arm where the amino acid will join. So it will join with this OH group. So COOH of the amino acid will form the bond here. This is the anticodon loop. Here you are going to get the anticodon. Now, bache, this is your DHU loop. Why it is DHU? Because here you will see some unusual bases as well, right? Dihydrouridine. You are going to find one unusual base here, that is dihydrouridine. Okay, that's why this is known as DHU loop. Okay, and here it is T-Psi. This is Sina, so it is pseudouridine. It is what? It is pseudouridine. So it is also the unusual base. Usually we talk about adenine, guanine, cytosine, thymine in DNA and uracil in RNA. But here they are what? They are what, Sabha? They are the unusual bases. So DHU is dihydrouridine and the Psi here is pseudouridine. Okay, these are also two different arms. So here when you talk about these arms, D, you know what is my trick to remember? D for down. D for down. This is my way to remember this. D for down, you are feeling down, you are feeling low. What do you need? You need energy. At that point of time, what do you need? You need energy, yes or no, Shrutik? Shrutik, Trisha, Sabha, everyone. Yes or no? When you are feeling low, you are feeling down, you need charging. Isn't it? You need charging. You need energy. You need charging. You are low, you are down. You need to charge your battery. Isn't it? Isn't it? This is my way to remember. So when I'm feeling down, when I'm feeling low, right, I need charging. And for the charging, what is required? Enzyme is required. Right? What is required? Enzyme is required. Right? So this DHU loop is going to bind with the enzyme which will help in the charging of tRNA. Are you getting it? That's my way to remember this part. This DHU loop, D means down. Someone is feeling low, right? We need charging. So we need charging. For the charging, enzyme is required. So this arm, this loop is going to bind with what? It is going to bind with the enzyme. And which enzyme is there? Your tRNA amino acyl. Yes, your tRNA amino acyl. Can you tell me the name of that enzyme? Can you tell me the name of that enzyme? Amino acyl. Yes. Anyone here in the class? tRNA amino acyl, the name of the enzyme. Synthetase, amino acyl synthetase. Okay, okay. So that's what you guys can, okay, that's what you people can write here. So this loop will bind with enzyme. Which enzyme it is? Amino acyl tRNA synthetase. Amino acyl tRNA synthetase. So do not make the mistake here. It is synthetase, not synthase. Synthase and synthetase, these are two different enzymes, right? In one ATP is involved, in another ATP is not involved. Okay, so it is amino acyl tRNA synthetase enzyme with which this DHU loop will bind. So okay, this one with the mRNA, this one with the amino acid, this one with the ribosome, this one with the enzyme. So obviously T psi C, it will bind with ribosome. It will get attached to ribosome. So that's what you need to remember. Okay, that's what sunlight you need to remember. Is that clear? Sure? Sure, bache? Sure? So this enzyme here is important for the charging of tRNA. This is important for the binding with the ribosome. This is important for the binding to mRNA and to the amino acid here. So this CCA point, the arms, that which one will join with which part, these two are the most important questions from tRNA. Okay, and if you look at this, it is actually the variable arm. What's that? It is a variable arm. But its function is not so clear. It can have different bases, like this arm is having different number of bases, and on the basis of this variable arm we divide tRNA into two classes, class 1 tRNA, class 2 tRNA. That depends upon this variable arm. Okay, that depends upon this variable arm. Clear, clear, clear? So that's what you need to keep in your mind. So here also you guys can see the structure of tRNA. Okay, here you guys can see the structure of, okay, tRNA. Fine, fine. So this is your D loop. So D means down. So this is going to join with the enzyme, with the ribosome, with anticodone, and here the amino acid. So C, CCA. Okay. Done. And this part is that variable loop or the variable arm. It is also having that unusual basis. Clear? Sure? Are you sure? So this is all about the different types of RNAs. Okay, this is all about the different types of RNA. Any doubt? Any doubt? So now let's start our actual part, that is your transcription. Okay, that is your transcription. Okay, so let's start with the basics first, bache. Let's start with the mechanism and then finally the NCERT reading. Okay, then we'll go for what? We'll go for the NCERT reading. Okay, so what is this transcription? It is the copying of information, especially the genetic information. It is the copying of genetic information from one strand, okay, of DNA to form mRNA. It is the copying of genetic information from one strand of DNA to form mRNA. So please focus on this word. Okay, so it is one strand of DNA. That's important. It is the one strand of DNA. Clear, bache? Clear, bache? This point is important. Now, see, okay, so what do you think that from where the transcription is going to start? Like when we were talking about the, when we were talking about the replication, we discussed now that replication is going to start from, yes, the replication is going to start from the origin of replication. That is what we were that's what we have discussed, isn't it? That's what we have discussed, isn't it? But when it comes to the transcription, obviously it is not going to start from any point. Okay, it is also not going to start from any point. So this is what you need to remember. The transcription starts from transcription unit. It is going to start from transcription unit. Clear, bache? So that transcription unit, it includes three things. It is having a promoter, the terminator, and the structural gene, right? And the structural gene. Clear? So promoter, terminator, and the structural gene is important, right, bache? So transcription is going to start from transcription unit, and transcription unit include these three things. Okay, it is going to include these three things, but there are more things that we need to remember. And here also I'm mentioning this point. In eukaryotes, your transcription site is nucleus. Okay, it is nucleus. That's important. In prokaryotes, that site is cytoplasm. Fine, that site is cytoplasm. Okay, so now one very important point related to this. See, earlier it was believed that DNA is just making the proteins, hai na? The DNA is the part which is only making the protein, it is believed, but actually it is not like that. Now come to the NCERT. Okay, I will tell you a few things from NCERT, and then we will start this process in detail. I will tell you about the mechanism. So here in this topic, we will also talk about the prokaryotic transcription. We are also going to talk about the eukaryotic transcription. So we are going to consider both. We are going to study both. Okay, that's what you have to keep in your mind. So here you guys can see, it's very simple. The process of copying genetic information from one strand of DNA is known as transcription. Okay, and on what basis this transcription is dependent? See, complementarity. The principle of complementarity governs the process of transcription, right? That is something very, very, very important. Nandini, please highlight this. Okay, please highlight this, that it is the principle of complementarity that is going to govern the process of transcription. The process of transcription, the complementarity. Now, what is the meaning of this? You know it very well, right? That if adenine is going to find bound with thymine, cytosine will form bond with guanine. But now, in RNA, we know that thymine is absent, and what is present? Uracil is present, isn't it? Uracil is present. So here also, the DNA is going to follow the rule of complementarity, but in a different way. Let's say this is your DNA. Just imagine this is your DNA. Right now I'm not specifying the 5' and 3' and randomly I'm teaching you this. A, T, C, G, T, T, A, C, G. Let's say this is DNA. Now you have to copy the information. You have to make the mRNA. Okay, Shiva Kumar, you have to make the mRNA. So now when it is G, you will get the C here, complementarity. When it is C, you will get the G here, complementarity. But now you know that adenine is complementary to thymine, but in RNA, the thymine is absent. So of course, at the place of A, there will be the U. There will be the U. Here also, at the place of A, there will be the U. At the place of, now this is thymine. Thymine, complementary to thymine is adenine. So here you will get adenine. C, G, G, C. So TT means AA. AA means UU. Like this. Like this. So in RNA, at the place of T, there will be the U. That's what you need to keep in your mind. So ultimately, it is also following the principle of complementarity. Okay. Okay. So that's why they have mentioned it here. Except the adenosine now forms base pair with uracil instead of thymine. Okay. Instead of thymine, uracil, adenine will form. Yeah, adenosine will form bond with the uracil. Okay. Okay. And in the process of replication, you know that total DNA of organisms get duplicated. Both the strands will open up. They will behave like a template. They will be copied. But in the transcription, only a segment of DNA, only a particular segment, single strand and a segment of DNA. See, it's not like that the complete strand of DNA will be copied. No, it is just that transcription unit. Okay, and from that transcription unit, sunlight, only one strand, only single strand will be copied. Is that clear? So here you guys can see that right? Unlike in the process of replication, which once set in the total DNA of an organism gets duplicated, in transcription, only a segment of DNA and only one of the strand is copied into RNA. Only one of the strand. Okay? Okay? So from that single strand, only a stretch of DNA will be copied. That's what you need to keep in your mind. So this necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. So that's what we need to discuss. Okay? Okay. Sure. Now I will tell you about the cistrons as well. So why both the strands are not copied and all, this is what I need to discuss with you all. But before that, just a minute. Let's start it from this part. Okay, let's start it from this part and then I will tell you about the transcription unit. So see, you know that it is the gene which is the functional unit of inheritance. Am I right? You know that it is the gene which is the functional unit of inheritance. Isn't it? Isn't it? Yes. Gene is the functional unit of inheritance or heredity. So there is no ambiguity that the genes are located on DNA. Right? Though there is no ambiguity that the genes are located on DNA, it is difficult to literally define a gene in terms of DNA sequence. No doubt we know gene is the unit of inheritance, but it's difficult to define it as in the terms of DNA sequence. So DNA sequence is also, right? Ultimately, here what are we discussing? We are saying that your DNA, right? Initially, this is what we used to say, that DNA, the segment that is getting copied, it is gene, because this is gene is the unit of, right? It is the gene which is the unit of inheritance, but it is difficult to define gene in terms of DNA. Actually, bache, your gene or your DNA is not just making the protein, it is also making tRNA, it is also making ribosomal RNA, it is also acting like a unit, like it is also available for the mutation, it is also available for replication, it is also available for recombination. Isn't it? Isn't it? Yes or no? Isn't it? Have a look here now. So DNA sequence coding for tRNA or RNA molecule also define a gene. You cannot just say that gene is that DNA sequence which is going to form protein. No, it's not like this. Nandini, it's not like this. So DNA sequence is also coding for tRNA. It is also coding for rRNA. It is also defining a gene when they are coding for tRNA, RNA. Moreover, that segment of DNA is also acting as a segment from where replication can start. That DNA segment is also the part which can undergo mutation. Okay, the DNA is also available for the crossing over. So you can't just define it in the sense of the gene, in the terms of the gene. Are you getting it? Are you getting it? So by defining a cistron, as a segment of DNA, because here you are talking about what? Here you are talking about what? You are saying that DNA is forming mRNA and that mRNA is forming protein. That's our central dogma. That's our flow of information. So, bache, the stretch of DNA that is making protein, right? That is used for making protein. That is going to form the protein. You are going to call it as cistron, right? It is just to avoid the confusion, right? So, I am repeating this part again. Hope you understand that DNA, its segment can be used for making tRNA also, for RNA also. It can be the part of replication unit also. It can be involved in the recombination also, for the mutation also. But when we are specifying that the segment of DNA, it is making the protein, so we will use that segment. We will use the word cistron here. Which word are we going to use here, bache? Which word are we going to use here? The word is cistron here. Right, the word is cistron here. Okay. So cistron is so structural gene and transcription unit. It is basically cistron. So that's what we need to discuss. So now we will continue this part. Okay. Now we are going to continue this part. So just have a look of this wonderful diagram. Right? Isn't it? This diagram is wonderful. It is showing a plant cell. Right? It is showing a plant cell. So it is showing the nucleus of that plant cell. Right? So here in this nucleus, this is that linear DNA which is forming the mRNA. So this mRNA is undergoing the processing. It is coming out It is used for the protein synthesis in cytoplasm. So this process, this diagram is repeating, this diagram is explaining everything. You can have a look. Okay, you can have a look. Done, bache? Yes. Is it clear? Yes or no? Is it clear? Sure. So let's start this part. Fine. Let's start this part. So please pay attention. That's important. Or you can say that that's very important. This is a stretch of DNA. That's your strand having polarity 3' to 5'. Another strand having polarity 5' to 3'. As I said in a transcription unit, you will see the promoter. You are going to see the terminator. And here in between, this portion is what? This portion that we are going to copy is your structural gene. What's that? It is your structural gene. Are you getting it? This central portion is what, bache Nandini? It is the structural gene. Now, here in the structural gene, because it is a gene is the basic unit of inheritance. It is ultimately the DNA, so having two strands. So this strand having polarity 3' to 5'. This strand having polarity 3' to 5'. This strand is known as your template strand. Right, it is known as your template strand, and the strand opposite to it is known as your coding strand. Then it is which strand? It is the coding strand. Are you getting it? It is the coding strand. Let me explain this again. See, when we were discussing the DNA replication, that's what we were doing. Just a minute. Array 3', 5', 3'. Right? Basically, it is helical. No, just a minute. It is a 5', 3', 5', 3'. If I am taking it in this way, right? That is this DNA that we have. So when we were trying to open it, you know that we were getting the replication fork. Or when we were getting that replication fork, we know that like this strand is what? It is 3' to 5'. It is 5' to 3'. Okay, this is 3' to 5', 5' to 3'. So opposite to this 3' strand, we were getting a leading strand. We were getting that template strand, we were getting that continuous strand, the leading strand. So here, Shrutik, we were discussing that the template strand or the parental strand having polarity 3' to 5', opposite to it, leading strand will form, right? Opposite to it, leading strand will form. That's what we were discussing, right? Now here also it's all about the polarity. You really need to know about the polarity. See, look at the direction of the arrows, 5' to 3'. The direction of the arrow is showing synthesis here. Do not ever get confused. I always told you that synthesis will be in 5' to 3' direction. So this is 5', this is 3'. So this arrow is showing what? It is showing that synthesis. And here 5' to 3'. This is showing that. Okay. Here also it's like that. So these arrows are showing what? They are showing the direction of synthesis. But what you need to consider here, that when it comes to the transcription, we are only going to take one strand of DNA to copy the information. Why? Because your mRNA is a single stranded. It is not double stranded. But if I will copy both the strands, then I will get the mRNA, and these mRNAs, I will get two mRNAs, and both of them will be complementary. And if both of them will be complementary, they will form hydrogen bonds. If they will form hydrogen bonds, so during Translation, right? I have to spend more energy to open this mRNA. I have to spend more energy to copy that information, and ultimately it is going to give us that same protein. And moreover, if we will read it in different directions, then different sequence of amino acid can form different proteins. So it is not beneficial for us. Okay, it's so mRNA is single stranded. Why it is single stranded? So that it can act as a template for making the protein. We don't need double stranded mRNA. We don't have that much energy to spend here. Already it's very energy draining process, energy taking process. So we don't need that much of energy. So if we will copy both the strands of DNA, they will give us the mRNAs which are complementary to each other, which will result in the formation of double-stranded mRNA. So if you have completed your syllabus once, then you know that we have studied it, such kind of things, double-stranded mRNA in your biotechnology. Remember we talk about the process there, na, RNA interference. In RNA interference, Subhashini, we discussed this. Okay, there I will tell you this. But here, remember, only one strand will be copied, and I told you the reason as well. So the strand having polarity. Okay, remember these points, these bullet points, right? So strand having polarity 3' to 5' will act as a template strand. Okay, it is going to act as a template strand. Okay, so template strand is having another names as well. Do you know that? Template strand is also known as non-coding strand. Template strand is also known as anti-sense strand. Do you know that? It is also known as minus strand. These are the names. These are the names of the template strand. Do you know that? It is no doubt, it is template. Template means it is available to copy the information, right? But the coding strand is the another one. So we call it as non-coding. We even call it as anti-sense and minus strand. Okay, so you guys can get confused here, because when we hear something like this, the anti-sense, so we are like, nahi, it is not going to copy the information. No, it's not like that. But here this is the case. Okay, so template strand, non-coding, anti-sense or minus, and here the coding strand is also known as, you can write down, it is known as non-template strand. It is known as sense strand. It is known as plus or the positive strand. It is negative or it is the positive one. Clear? So do not ever get confused. Do not ever get confused. Now, when you are saying promoter means which is going to promote something. So it will be the one which will promote the transcription. Terminator, to terminate something, to stop something. So it is the one which is going to terminate, which is going to stop something. Clear, bache? So promoter will promote transcription, terminator will stop transcription, and here in between we have the structural gene that we need to copy. It is having that information. Okay. So when it is the template strand, this is the template strand, and that template strand will be copied. Okay. Now imagine your template strand is having polarity 3' to 5', and you know that whether it is DNA or RNA, it will be synthesized in 5' to 3' direction. So this is DNA. So complementary to it, RNA will form. That will having this polarity. Simple. So it's all the game of polarity here. Fine. So polarity here is very important. So any doubt here? Now, see, I just told you that when you are talking about the structural gene, it is that portion of DNA that you are going to copy that will form mRNA, and then it will form protein. Ultimately, your mRNA is going to form the protein. So this part here, okay, this part of structural gene here is something, okay, this part of structural gene here is something which will be copied, which will form the protein, right? It is going to do what? It is going to form the protein. Okay, so in the case of prokaryotes, okay, so because this is the part of DNA which is forming the protein, so can we use the term cistron here? Because cistron is that part of DNA which will form a polypeptide. Cistron is that part of DNA, Trisha, which will form a polypeptide. So can I use the word cistron here? Yes, of course I can. So, bache, in the case of prokaryotes, P for polycistronic. Your prokaryotes are polycistronic. And when you talk about the eukaryotes, right? When you talk about the eukaryotes, so it is going to be monocistronic. What's that? It is going to be monocistronic. Is that clear? Is that clear? Now, what is the meaning of polycistronic and the monocistronic here? Yes, what is the meaning of monocistronic and the polycistronic here? Again, bache, it's very simple. See, poly means many cistrons, many polypeptides from one structural gene. Mono means a single. So do not confuse it ever. Prokaryotes are polycistronic, but they are monoreplicant. Never ever confuse this part. You can include it in your notes. Prokaryotes are polycistronic, but they are monoreplicant. Eukaryotes are monocistronic, but they are polyreplicant. So please do not get confused. Cistron word is associated with the protein synthesis. Replicon word is associated with the replication. So in the prokaryotes, there is only one origin of replication. In eukaryotes, there are many. But in prokaryotes, when you are talking about one transcription unit, there can be many structural genes, right? One RNA polymerase can transcribe many structural genes. But in the case of eukaryotes, there is only one structural gene here. You will get that only. Okay, so initially you will get only one polypeptide. This part is clear. Is it understood? Yes, this part is clear. Is it understood? So structural gene is having the polarity 3' to 5'. So it is the template strand. So I hope you understand this. I hope you understand this. Sure? So it's all about the polarity, students. It's all about the polarity here. Are you sure? If there is any doubt, do let me know. If there is any doubt, do let me know. But poly means many. Cistron is the part of DNA which is related to the polypeptide synthesis. So when it is a structural gene, actually the story is going to be like this here, right? When it is a prokaryote, the story is going to be like this. One RNA polymerase can transcribe many structural genes. There will be structural gene 1, structural gene 2, structural gene 3, right? So during one, right, that RNA polymerase will give you three different mRNAs that will give you 3 different polypeptides. You discuss that in Operan, Nandini. You discuss that topic in Operan. But in the case of your eukaryotes, you will get only one structural gene, and because of that, you will get only one mRNA, and then not exactly one polypeptide, because here also the split gene concept comes, but actually this is the case. So here you will get only one structural gene. Is that clear? You're going to get only one structural gene. Done? Sure? Doubt? Any other doubt? So, okay. So, let's have a look. See, promoter, RNA coding region, terminator. So, here you will see transcription terminator site. But right after the promoter, this information is important, students. This site here is actually the transcription start site. We also call it as start point. What we used to call it? Start point. So here, this is the non-template strand and the template strand. So ultimately, don't be like that if it is like this, right? If it is like this, don't be like, this upper one is going to be the template. No, always focus on the polarity. Always focus on the polarity. So the polarity is 3' to 5', then that will be our template strand, and another one will be non-template, and it is also known as the coding strand, right? It is also known as the coding strand. Is that clear? Understood? Is that clear? Understood? So you can see another example as well. So no doubt in this process, we'll be discussing Tata box, this box, that box, but as of now, just keep it in your mind. Fine? Just keep it in your mind. Doubt, bache? Any doubt? Sure. So let's proceed. So first of all, let's start it in the case of prokaryotes. Fine, let's start it in the case of prokaryotes, and remember there will be 3 steps. First is initiation, that is you have to start this process. Second is elongation, you have to elongate, you have to synthesize mRNA, and then you have to terminate this. Then comes the termination. These are the 3 steps of the prokaryotic transcription. Clear, bache? These are the 3 steps of the prokaryotic transcription. The initiation will be there, the elongation will be there, and then comes the termination. Then comes the termination. Clear, bache? Clear, bache? Are you sure? Are you sure? So, because it is prokaryotic transcription, so what are you going to talk about? You will discuss RNA polymerase. It is also known as DNA dependent RNA polymerase. What is it? It is DNA dependent RNA polymerase. It's DNA dependent RNA polymerase. Another thing that you are going to discuss here is your sigma factor. And then comes your rho factor. Okay, so RNA polymerase, sigma factor, rho factor, that's what we need to discuss here. Okay, that's what we need to discuss here. Okay, but you leave everything. I'll ask you few basic questions, few general questions, and on the basis of that, we will discuss this process. Okay, then we will discuss this process. I'll clear few points. Okay, let's start like this. I told you that there is the promoter region. I told you there is the terminator region. That's what I said. So if I am saying that, that's known as RNA polymerase. Okay, that is known as RNA polymerase. Are you getting it? What is it? It is known as RNA polymerase. And on the other hand, bache, on the other hand, when you are talking about eukaryotic transcription, let me tell you this thing here only so that you don't get confused. Okay, so when it is the eukaryotic transcription, the site is nucleus, steps are same, initiation, elongation, termination. But here you will talk about RNA polymerase 1, you will talk about RNA polymerase 2, RNA polymerase 3. That's what we have to discuss here. And here we are not going to discuss no sigma and rho factor here. No sigma and no rho factor here. No sigma and no. Trisha bache, see, this is what you need to observe. I'm saying that in prokaryotes, that's what you need to understand. In eukaryotes, 3 steps are same. Site is nucleus. 3 RNA polymerases are going to play the role here. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. And no sigma and no rho factor will be there in the transcription of the eukaryotes. Instead of that, there are another transcription factors. This transcription factor D, A, B, G, E, H, like this. Okay, no doubt we are not going to discuss it. We are not going to discuss it because it is not included in NCERT, but this is just for your information. Okay, so in prokaryotes, sigma and rho factor will be discussed. In eukaryotes, there is no such thing like sigma factor and the rho factor. Okay, here you will be discussing transcription factor D, A, B, G, H, E. Done? Sure, this part clear. This part clear. So now we are talking about the prokaryotes, and as I said that there is the transcription unit and there is only one RNA polymerase. But why do we call it as DNA dependent RNA polymerase? Why are we saying that it is DNA dependent RNA polymerase? Because obviously it will use this DNA template and then it will form the RNA. So it is DNA dependent RNA polymerase. Clear bache? Clear bache? Yes or no? Yes or no? So now I told you there is one more factor which is known as sigma factor. In the prokaryotic transcription, that's what I said, we have one factor known as sigma factor. Bache, S for sigma, S for start. S for sigma, S for start. Yes or no? S for sigma, S for, tell me students, S for sigma, S for start. So it is something which will help in starting the transcription. So what is the role of the sigma factor? But you see, you have this enzyme known as RNA polymerase. Again, in the DNA replication also we were imagining everything, na? Story was going on. है ना? In our background a story was there कि okay, that's how it works, this is how it is going. So imagine there is one RNA polymerase. RNA polymerase is ready to start its work, but RNA polymerase don't know where to go. RNA polymerase is not aware where to join. Okay, RNA polymerase can start its work by its own, but it doesn't know where to go. So sigma factor will be a savior here. Sigma factor will come and will tell RNA polymerase that I will take you to the promoter site. I will tell you where you have to bind. I will tell you, right? I will help you there. Is that clear? Is that clear? Yes or no? Is that clear? Yes or no? Sure? Sure. Are you sure? So the story is not simple. I told you that this is a kind of promoter region. If it is a promoter region, what that promoter region will contain, bacha? Of course the nucleotides, of course the nitrogenous bases. And I told you, here you are going to get a start point. Here you are going to get a start point, the actual point from where the transcription will start. The actual point, Priyanka, from where the transcription will start. Now it is something very easy. Okay, it's something very easy. That's what you need to remember. Let's say this is your start point. Let's say this is your start point. If you are going to this direction, up, above, upstream. If you are going to this direction, then you will use the word downstream. Just keep it in your mind. Just keep it in your mind. There is a point of reference. Let's say I am the point of reference. If you are moving towards my right side, I'm saying you are moving upstream. If you are moving towards the left side, I'm saying what? I'm saying the downstream. So here, if something is moving like this, up, down, this point clear? So you are going to take the reference of a particular point, and with that reference, that's what you are going to say. This is upstream, this is downstream. This is upstream, this is downstream. Understood? Yes, everyone. Understood? Sure. So now let's say if I'm taking this point of reference, so this side will be the upstream, this side will be the downstream. Yes or no? Priyanka, if I'm taking this as the reference point, right bache Shrutik, so this part will be the upstream, this part will be the downstream. Yes or no? Guys, be quick. Be quick. Of course. So let's say here it is the structural gene. This is your template strand. This is your structural gene. So this portion is going to be the upstream. This portion is going to be the downstream. Of course, yes. Of course, yes. Right? So now I told you that it is the sigma factor which will guide the RNA polymerase, will help that RNA polymerase to bind with the promoter. So in the NCERT, it is written in a very simple way. In the NCERT, it is written in a very simple way, but actually the story is not like that. Other proteins are also going to play the role, but that's not the part of your syllabus, so you are safe. So, but remember, when it is the promoter, in the promoter, upstream to that promoter, upstream to that promoter, upstream to that promoter. How? Right, upstream to that promoter. Upstream to promoter means towards that side, right? So it is actually before 10 nucleotides or 10 nitrogenous bases, right? Like let's say this is a start point, so leave 10 nucleotides or 10 nitrogenous bases here, then you are going to find something known as your what? Known as your Tata box here. Okay, known as what? Known as your Tata box. Clear, bache? Known as what? Known as your Tata box. So it's TATAAT. That's why we are calling it as Tata box. Like this is the start point, or this is the promoter here, right? This is the start point and this is the promoter here. So if I'm considering this as the start point, so 10 nucleotides upstream to that start point means somewhere here in this promoter, you people are going to get Tata box. right? You people are going to get Tata box. So upstream to start point. So upstream to start point, you know the meaning now, right? You all know the meaning now, isn't it? Isn't it? So 10 nucleotides above this start point, what will you get? You will get the Tata box. What will you get? You will get the Tata box. So it's TATAAT. It's TATAAT. So here it's P for prokaryotes, P for priv now box. Priv now box. Okay, it's priv now box in the prokaryotes. Clear, bache? Clear, bache? Clear, bache? And even one extra point I'm telling you here, that even in eukaryotes also, you will see such Tata box. In eukaryotes also there is such Tata box, but that Tata box is known as Goldberg-Hognes box. Okay, and it is having near about 7 bases, so it's TAT this. Okay, so in prokaryotes, P for priv now, P for prokaryotes, TAT, this sequence is present, having 6 base pairs. But here in eukaryotes, okay, here in see, this is TATAAA Goldberg-Hognes box. So it is also upstream to the start points, near about 20 nucleotides above. Clear? Upstream, downstream, clear? Upstream, downstream, clear? Yeah, Sabha, you can also use the same language, that upstream to the structural gene. That is also fine. Okay, that is also fine, but then number of bases will change. Fine, then number of bases will change. Okay? Clear? Sure? Tell me. Sorted? Yep. Is it sorted? So now sigma factor, right? Sigma factor is like a mentor. Sigma factor will pick up that RNA polymerase, will tell that RNA polymerase, this is the promoter, this is that PrivNow box, this is the Tata box where you need to join. And why Tata box? Because it's easy, right? See, bache, your DNA is double-stranded. Okay, your DNA is double-stranded. So here, right, when it is the Tata box, so here it will be easy for RNA polymerase to break that hydrogen bonds. Okay, because in adenine and thymine, like let's say if this is the Tata box, so complementary to it A will be there, complementary to it T will be there. So that bonds are easy to break in comparison to G and C part. So that's how, so sigma factor will pick up, will bring it to that promoter side, to near to that Tata box, so that it can break that hydrogen bond, so that it can open up that DNA helix. Is that clear? Is that clear? Right? Is that clear? Less energy will be used. So this is very important, and it was a question in NEET 2020. Right? It was a question in NEET 2020. The question was that in transcription, which enzyme is going to open up the DNA double helix? Which enzyme is going to open up the DNA double helix? And you know what came in our mind? Even I'm talking about myself. Now, when I read that question for the first time, I was like, we have to open up the DNA double helix, right? We have to open up the DNA double helix. So it is the helicase, right? Usually we do things like that, na? So I missed that part, that in transcription they are asking. So that's why we always used to say that you need to read the questions carefully. Okay, you need to read the questions carefully. Fine. So in transcription, it is the RNA polymerase which is going to open up the DNA double helix. Is that clear? Is that clear? Sure. Sure. Are you sure? Up to this part all clear? Now bache, now always remember, see, wait, I'll show you this part. This is for the eukaryotes, this is also for the eukaryotes. Yes, always remember, no doubt, here in your promoter, right, here in your promoter, RNA polymerase will bind with the help of your sigma factor, but ultimately the transcription, the mRNA synthesis will start from this starting point. It is going to start from this starting point. That's what you need to keep in your mind. Okay, it is ultimately going to start from this starting point. Okay. Okay, from here the mRNA formation will start. And moreover, let me tell you one more thing. Like in NCERT, we explain it in a very simple way, that RNA polymerase will come and then it will start the protein mRNA synthesis. It's not like that. Initially the mRNA that forms, it is like here, if it will start, it will form a short stretch of mRNA and even it gets removed, something like that. And then finally it start making that proper mRNA. Even this is also the story, this is also the case. But theek hai, again, let's not discuss that in detail. So remember, sigma factor will bring our RNA polymerase here, and once that RNA polymerase is comfortable, once that RNA polymerase be like, yes, now I'm happy here, I can just start the transcription, then sigma factor will say bye-bye to that RNA polymerase. Okay, it's not like that RNA sigma factor will stay with that RNA polymerase. It's not like that. Okay, that sigma factor will stay with that RNA polymerase, will be with that RNA polymerase for the elongation. No, once RNA polymerase adapt here, once RNA polymerase start the proper mRNA synthesis, sigma factor will leave the RNA polymerase, and then it is the RNA polymerase which will slowly unbind DNA double helix and will slowly forms the mRNA. So that's how the initiation and the elongation will go. Fine, Trisha? Fine, Trisha? Is that clear? These are very basic points but are very important points. So see, sigma factor is bringing the RNA polymerase here, right? RNA polymerase is unbinding the DNA double helix in transcription. So once it will start the mRNA synthesis, sigma factor will leave it. Sigma factor will leave it. Now it is synthesizing mRNA, synthesizing. Now there comes the terminator. There comes the terminator. So terminator is the one. See, ultimately terminator is also the part of DNA, am I right? Ultimately it is the terminator is also the part of DNA, am I right? Yes, Trisha, Shrutik, Sava. Terminator is also the part of DNA. So obviously there also we are having some bases, right? We are going to see some bases here. We are going to see some nitrogenous bases here which are giving a signal, now stop the transcription. Okay. So here which factor do you need? You need the rho factor. So if S for start, so obviously rho factor is going to stop the transcription. Rho factor is going to stop the transcription. And we also have rho independent termination of mRNA. Okay. We also have rho independent termination of mRNA. Means it's not mandatory that always rho factor is going to, terminate this. Here some nonsense bases will come. mRNA will get separated with the help of rho factor, but sometimes it is also rho independent termination, right? And in rho independent termination, a loop like structure will form which will separate mRNA from DNA. right? Which will separate mRNA from DNA. Are you getting it? Which are going to separate mRNA from DNA. Is that clear? So that is for the prokaryotic transcription. That is for the prokaryotic transcription. Okay, that is for what? For the prokaryotic transcription, and that's what you need to remember. Now, bache, I will ask one more question here. May I? So this is a random sequence. Okay, this is a random sequence. So tell me here which one is your template strand. Tell me here which one is your template strand. Which one is your template strand? Tell me, bache, tell me. 3' to 5', right? 3' to 5'. 3' to 5', am I right? Yes? Okay, this is the template strand. So complementary to it, see T, A, G, C, G, C, G, C, T, A, T, A, and the T. That's DNA. These are the sequences. Now you know that we are going to copy this part. So now tell me the sequence which is present on mRNA, and look at the polarity here. It's 3' to 5'. So obviously here it is 5' to 3'. That's how the synthesis will be. Synthesis is always in 5' to 3' direction. Okay. So now here it is A. Tell me the base is present here on our RNA. Quickly write down. This is the sequence on DNA. You have to tell what will be there on RNA. What will be there on RNA? Quickly, everyone. Quickly. Write down the sequence. Quick. Even I will write it. Right? Very good, Nandini. Very good. So that's your mRNA, right? That's your mRNA. So now when you look at the sequence, you have copied this particular strand of DNA. You have copied this particular strand of DNA when you look at the sequence of mRNA. That's the sequence. Now compare this sequence with the coding strand. That's your coding strand, right? That's your coding strand, right? Compare the sequence, Ritik. Compare the sequence of mRNA and coding strand. Bache, it's same. The only difference is at the place of thymine, uracil is there. Rest, it's all same. Right? Rest, it's all same. It's all same. Even the polarity, it is 5' to 3'. It is 5' to 3'. Only the difference is at the place of T, here you have the U, uracil is there. So that's why, because mRNA is having ditto information written on coding strand, that's why you're calling this particular strand as coding strand. mRNA is having the same information just like this strand of DNA. That's why we are calling this strand as coding strand. Okay, it is the coding strand. So if in the paper, sometimes they, if you will solve previous year questions, you will get it. They will give you the stretch of DNA, that this is the sequence present on the DNA. What will be the sequence on RNA? Obviously, in that case, you need to do the smart work. Just look at that coding strand and it will be same like the coding strand, even the polarity 5' to 3'. Only at the place of thymine, just put the uracil. Only at the place of thymine, you have to put the uracil. So be smart. Okay, be smart. So this is about the prokaryotes. What is it? This is about the prokaryotes. Now, when you are talking about the eukaryotic transcription, it's little different. in the case of eukaryotic transcription, first of all, it is in the nucleus, right? I have repeated it so many times. It will be there in the nucleus. Now, not just in nucleus, here also you will see another thing. Do you know that? You are going to see another thing as well. Okay, here when you are talking about the eukaryotic DNA, see bache, in the case of prokaryotes, remember this thing, in the case of prokaryotes, you talk about operants. Why? Because prokaryotes are your polycistronic. Here we discuss something called operants. We will discuss it, okay, tomorrow. In the case of eukaryotes, we do not talk about the operants. In the case of eukaryotic DNA, you are going to study about the concept of split genes. That's important. Please listen to me carefully. You will discuss the concept of split genes. In the eukaryotes, after transcription, you will also talk about RNA processing. Okay, you are going to talk about what, Nandini? You are even going to talk about the RNA processing. Clear? Clear? You are even going to talk about what? You are going to talk about the RNA processing as well. So just look at this. In the eukaryotic DNA, what do you have? You have the split genes. Now, what is the meaning here? What is the meaning here? Like, when you look at this DNA, when you look at this DNA, okay, this DNA is having the coding region as well, the non-coding region as well. In the DNA of the eukaryotes, there will be the part which is having the coding region. Coding region means it is providing me the information to form a protein, but it is just like a kind of it is the interrupted information. What type of information it is? Kind of interrupted. Wait, I'll show you. Like, see, here in the DNA of your eukaryotes, one part is the coding part, another part is the non-coding part. Then again you will see the coding region, the coding part, then you will see the non-coding part, right? What are you going to see? You're going to see the non-coding part as well. Such kind of things will be there. Okay, such kind of things will be there. The coding part, the non-coding part, the coding part, the non-coding part, like this. So ultimately the coding region, when it is the coding region, you use the word exon for it. Which word are we going to use for it? Exon. And when you are talking about the non-coding region means it is the one which will not provide me the information to form the protein. Okay, Sabha, it is the part, Nandini, which is not going to tell me that which protein do I need to make. It will not tell me about the protein. And here it is going to tell me about the protein. That's the point. Clear? That's the point here. Are you getting it? Yes? Are you getting it? I hope you are understanding it. So these are the split genes. Okay, it is kind of evolutionary. It is evolutionary. Okay. So genes which are having non-functional part with functional part, they are the split genes. Okay, genes that are having, it's given in NCERT, functional part and non-functional part. Functional part means the coding part, non-functional part means the non-coding part. They are the split genes. Clear, bache? They are the split genes. Is that clear? Is that clear? Yes or no? Yes or no? So when in the eukaryotic transcription, right, when there is the, see, in the eukaryotic transcription, the first point is RNA polymerase 1, RNA polymerase 2, RNA polymerase 3 will be there. These three enzymes will be there for the transcription. So it's RNA polymerase 3. 3 means 3, T. So it will form your tRNA. 2 will form your mRNA. And 1 will form your rRNA. This is in general. This is how you can remember. 3, tRNA, second will form mRNA, and 1 will form your rRNA. There are exceptions, but that's how you have to remember. That's how you have to remember. Yes or no? Yes or no? So these three enzymes are used to make the different types of RNAs that we need. Let's say if it is mRNA, you need RNA polymerase 2. Okay, you need RNA polymerase 2. So that RNA polymerase 2 is going to transcribe the structural gene, and this structural gene is having coding as well as non-coding region. This structural gene will have coding as well as non-coding region. So don't you think, bache, don't you think that the mRNA that I'm going to get, if DNA is having the split genes, if DNA is having that coding region, yeah, functional region, non-functional region, functional region, non-functional region, functional region, non-functional region. So don't you think the mRNA that I'm going to get, it will also have the same thing? Let's say this part is the coding part. And this one is going to be non-coding. Non-coding. Getting it or not? So that's what we see in eukaryotes. So in eukaryotes, because this process, this entire process is going on in nucleus. This process is going on in nucleus. So DNA is forming this mRNA. DNA is forming this mRNA, yes or no? And this mRNA is not the mature mRNA. It is having coding, non-coding, coding, non-coding, coding, non-coding region. So you used to call it as immature mRNA. Okay, or precursor mRNA, or heterogeneous nuclear mRNA, also known as hnRNA. Are you getting my point? hnRNA. So what type of mRNA it is? What type of mRNA it is? It is going to be your immature mRNA, precursor mRNA, heterogeneous nuclear RNA, or hnRNA. hnRNA. Okay. And 3 polymerases are playing the role. I will tell you the exceptions of that. RNA polymerase 1 will form your ribosomal RNA. I already told you about the ribosomal RNA that you have. 5S ribosomal 5.8S ribosomal RNA, 28S ribosomal RNA, even the 18S ribosomal RNA. But here one exception is there. Your RNA polymerase 2, it is forming your mRNA as well. It is forming your 5S ribosomal RNA as well. That 5S ribosomal RNA is the exception. It will not be synthesized in nucleolus. It will be there in nucleoplasm, and RNA polymerase 2 is going to form it. It is going to form it. Clear? That is an exception, or obviously it is what? It is the PYQ. So that's important. That's important. Right? So now in my eukaryote, in the nucleus, that's what I'm going to get. Immature precursor heterogeneous nuclear hnRNA. So don't you think that if I have to send it from the nuclear pore complex, if I have to send it in cytoplasm, don't you think that firstly I need to make it mature? I need to make it mature, yes or no? Let's say winters are there. How many of you love winters? Tell me, how many of you love winters? Okay. One correction. This is RNA polymerase 2 will not form your 5S ribosomal RNA. My bad. It is your RNA polymerase 3. Okay, by mistake I wrote it like this. So it's tRNA and then your 5S ribosomal RNA. Yup. Then it's 5S ribosomal RNA. Clear, bache? Then it's 5S ribosomal RNA. Okay. Okay, so RNA polymerase 2 will form your mRNA, and you know that what type of mRNA it is? It is your immature, or you can say that heterogeneous nuclear mRNA. So, so how many of you like winters? Winters, that ma'am, we love winters. So in winters, if you have to go out, you cannot just go out in a t-shirt. Simply in a t-shirt or shorts. No, the temperature is low outside. You have to put that sweater. You have to put the cap. You have to take the shawl. Isn't it? It's like that. So now if I have to send my mRNA to the cytoplasm for the protein synthesis, I will not I'm not going to send it in this form. That, you are immature, just go there. Just go there, you will learn the things by your own. No, I have to make it mature first. So that's why in the case of eukaryotes, after the transcription, when it comes to the post-transcriptional changes, right? After transcription, the kind of changes that are going to occur here, it includes the RNA processing. So what is going to happen in eukaryotes? RNA processing. RNA processing will be there. What will be there, students? RNA processing will be there, right? So here I have my mRNA, which is having the coding regions and the non-coding regions. So let's say these are the non-coding regions. So for non-coding regions, what we used to call them as introns. Introns. What we used to call them? We used to call them as introns. So I have to do what? So obviously removal of introns is important. The removal of intron is important. And this removal of introns, it is known as what? Splicing. It is known as what? It is known as splicing. Okay? It is known as splicing. And for this, we need some proteins, spliceosome. We used to call them as spliceosomes. Clear, bache? What we used to call them? Spliceosomes. So in NEET previous year questions, right, you will check one question there. They have given you spliceosomes are found in, right, bacteria, fungi, yeast, okay, amoeba, bacteria, yeast, paramecium, like these kind of options are there. So you have to be very clear. When it is bacteria, it means it's prokaryotes. No RNA processing. No RNA processing, because it is taking place, right? Transcription and translation is taking place simultaneously in bacteria. Clear? Clear? So spliceosomes are the protein complexes which are going to remove these introns. Clear, bache? Spliceosomes are the protein complexes which are going to remove what? Which are going to remove these. Yes, bache? Which are going to remove what? Which are going to remove this? Tell me. The introns. Is that clear? Is that clear? Sure? Sure, sure, sure. Are you sure? So what will be there in the spliceosome? Can you tell me what will be there in the spliceosome? Yes? Actually, bache, this spliceosome is assembled. It is found in eukaryotes. It is assembled from small nuclear RNA. snRNA means small nuclear RNA. snRNA means small nuclear RNA. And let me tell you, your RNA polymerase 2 forms snRNA. RNA polymerase 2 forms snRNA. So snRNA means small nuclear RNAs, right? So spliceosome is the assembling of small nuclear RNA and the proteins. And there are some ribonucleoproteins. Small nuclear RNA and some ribonucleoproteins are present here. Small nuclear RNAs and some ribonucleoproteins are present here. They are forming your, yes, bache, they are forming your spliceosome. Clear? Clear, 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 clear. So ultimately what is happening, your snRNA and proteins, they are going to form the spliceosome. So for that, yes, you use the term right? Ribonucleoprotein complex it is, because it is having small nuclear RNA and protein. So we call it as ribonucleoprotein complex. What we used to call it? We used to call it as ribonucleoprotein complex. Let me write it like this, SNRNP. So small nuclear ribo, right? So it will be small nuclear ribonucleoproteins. Small nuclear ribonucleoproteins it is. Clear? So splicing, removal of introns, which is what is going to help here in the removal of introns? That is your spliceosome, right, bache? That is your spliceosome. Clear? So spliceosome is formed by the assembly of small nuclear RNA and the protein. It is formed by the assembly of small nuclear RNA and the protein. So it is forming snRNPs. So they are small nuclear ribonucleoprotein complexes. Okay, 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 okay. Sure, 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 sure. Right? So these are done, bache. So they are basically kind of SNRPs, right? You pronounce it as SNRPs. So is that clear? They are of different types. Fine, they are of different types. Okay, so small snRNPs, SNRPs, we pronounce it as SNRPs. So small nuclear ribonucleoproteins are present here. They are of different types. They will remove this intron. So intron removal is in different ways. Sometimes a lariat is formed, so that part is not required. So this is what you need to know about this. Okay, this is what you need to know about this. So different type of SNRPs are there. Here they will form the splices. So once the introns, they are removed, obviously introns means non-coding. They are not going to provide us the information for the protein synthesis. They are not going to provide us the information for the protein synthesis, isn't it? Isn't it? So after the splicing, what are you going to get? Right? Only exons are joined, right? What will we see? Only exons are joined, right? So this will give us a functional protein. Now this mRNA is also functional. This is also giving us the functional protein. So that is very important step of RNA processing. But next step, you know what? It is not just the splicing. It is not just the removal of intron. There will be the capping and tailing of RNA. Do you know that? There is also the capping and tailing of RNA. Capping and tailing means cap of your RNA is 5' end. Tail is your 3' end. Tail is your 3' end. So capping means at 5' end, one unusual base. At 5' end, unusual base, that is 7-methyl guanosine triphosphate. What is the name of the base? It is 7-methyl guanosine triphosphate. That's how we write it, na? 7-methyl guanosine. So it's triphosphate. Why am I saying it is unusual? Why am I saying it is unusual, Sabha? Because usually the bases which are present in DNA or mRNA, they are monophosphates. They have single phosphate. That nucleotides are having single phosphate. But here what will you see? You people are going to see 3 phosphates. So it's 7-methyl guanosine triphosphate. It's 7-methyl guanosine triphosphate which will be added to the 5' end. And when it comes to the 3' end, in a template independent manner, template independent manner, adenyl bases will be added. Right? Let me come to this part. Capping 5' end, tailing 3' end. Okay, capping 5' end, tailing 3' end. So when it is the capping, it's 7-methyl guanosine triphosphate. 7-methyl guanosine triphosphate. And here when it is the 3' end, 100 to 200 adenylated bases will be added. Clear, bache? They will be added, and they are going to they will be added in template independent manner. They will be added in template independent manner. Are you getting my point? In template independent manner. What is the meaning of template independent manner? To add the bases here, you do not need the reference of another DNA or the RNA. No, without any template, you are simply going to put the bases here. You are simply going to put the bases here, adenylated residues here. Okay, adenylated residues here. Okay, and this will be done with the help of PAP. PAP is your PAP enzyme. It is an enzyme, polyadenylate polymerase. Polyadenylate polymerase it is. Okay, polyadenylate polymerase it is. This is the PAP enzyme that will help here. And here which base will be added? 7-methylguanosine triphosphate. So in eukaryotes, right, removal of intron is splicing. With the help of spliceosome it will be achieved, and then capping and tailing will be done. What is the purpose of this capping and tailing? Why that capping and tailing is required? It protects mRNA from nucleases or RNases present in cytoplasm. So in cytoplasm there are certain enzymes. There are certain enzymes in cytoplasm. Your nuclease basically, which can digest the nucleic acid, which can digest the RNA. So because of capping and tailing, that nucleases cannot destroy the mRNA. Okay, it cannot destroy the mRNA. Are you getting it? It cannot destroy the mRNA. Fine. Fine. So that is the RNA processing that will be done in the eukaryotes, right? So I told you about the transcription. I told you about it in detail. I told you about the Privnov box, about the Goldberg-Hogness box, right? So remember in the case of eukaryotes, it is not sigma and rho factor. Sigma and rho factors are absent in eukaryotes. There are other transcription factors like transcription factor A, B, transcription factor T, A, B, G, E and H. But there is no need to remember. But yes, you have to remember no sigma, no rho. No sigma, no rho. So that's how it works. Clear, bache? That's how it works. And now just look at this diagram. Okay, look at this diagram. It is obviously because exon-intron concept is there. Because there is the concept of split genes. So definitely it is eukaryotic DNA. So it is not just having the Tata box in the, upstream to the start point. It is also having, let's see, the promoter sequence, the Tata box, and enhancer you will also see in this. Enhancer you are going to see, right? Obviously it will enhance your transcription. So look at this. So Tata box, this is transcription factor binding site. It is the image of eukaryotes, na? It is the image of eukaryotes. So transcription factor binding site is here, Tata box is here, and here you will see 5-UTR and 3-UTR is also there. What is this UTR? Untranslated region, right? It will not be useful for protein synthesis. It is untranslated. Okay, and then this is basically that this is untranslated region. So here you will see the translation initiation site for protein synthesis. Okay, so these introns will be removed, exons will be joined. So in this case, you will see, right, in the mRNA of eukaryotes, you will see 5-UTR and 3-UTR will be there. Untranslated regions are there. Untranslated regions are there. Okay, okay, so see this. So exon, introns, 5-UTR, 3-UTR. So introns will be removed, all the exons will be joined, that part will be translated then. Okay, so here it is showing the transcribed region, the region which is transcribed, but this is how it is. 5-UTR, and here you will see the Tata box here, the enhancers here. So enhancers are actually upstream to the Tata box. Enhancers are actually upstreams of the Tata box. So you can take the screenshot of this diagram. Okay. Done? Any doubt? Any doubt? So that's all about the transcription. So now let's read everything from NCERT so that we can cover NCERT highlights as well. And then, bacha, we will start the genetic code. Okay, the mutations of genetic code and then the translation. So by tomorrow we will finish this chapter. Tomorrow we'll cover translation, we'll cover operands, we'll cover human genome project. Okay, the mutations as well. Okay, so be ready for that. But as of now, if there is any doubt, do let me know and let's discuss it from NCERT, right? So that you can, grab it in a better way. Fine? Okay, so let's go. So see, why both the strands are not copied during transcription? Now I think you know the answer. Anna CM, I think you know the answer. Why both the strands are not copied here? Why both the strands are not copied here? See, if both strands act like a template, they would code for the RNA molecule with the different sequences, of course, right? Sequences are going to be different proteins you are going to get, right? Because they are saying being complementarity. Remember, the complementarity doesn't mean that they are going to be identical. Identical means same, right? Ditto, same. Complementary means if adenine is there, uracil will be there. If thymine is there, adenine will be there, like this. So both the strands, they will be different, okay? They will be different. Plus, they also can get, okay, they can also form the double-stranded mRNA. Done, bache? Done, bache? And these two strands, if even they will not form, even if they will not form your double-stranded mRNA, they will give us different types of proteins because the sequence of amino acid will be different. Fine. So they code for the proteins, the sequence of amino acid in the protein would be different. So one segment of DNA would be coding for two different proteins. We cannot afford that. One part is giving us two different information. Can't afford it. Hai na bache? So it can complicate the genetic information transfer machinery. That's why. And moreover, RNA molecule if produced simultaneously, they will be complementary to each other. So they will form the double-stranded DNA. Is that clear? Right? Okay, so again the translation will be prevented. So now the transcription unit, these three things are there. So what is the polarity of the template? 3' to 5'. What is the another name of template? Non-coding strand. What is the another name for template? Antisense strand. Minus strand it is. Okay, so there is a convention in defining the two strands of DNA in structural gene of a transcription unit. So you know that the two strands have opposite polarity and the DNA dependent RNA polymerase catalyze the polymerization in only one direction. In only one direction it will form mRNA and that direction is 5' to 3'. So that's why the template strand that we have is having polarity 3' to 5'. Important MCQ. Important MCQ. Clear bache? Clear bache? So other strand having polarity, this is going to be your coding strand. And the sequence of this coding strand, I told you this thing, right? Right, bache? I told you this thing. The other strand which has the polarity and the sequence same as RNA. Only one point is at the place of thymine, uracil is there. It is the coding strand. I have given you the example as well, isn't it? It is the coding strand then. It is the coding strand then. So strangely, this strand which doesn't code for anything is referred to as coding strand. Why? So obviously it can confuse you, isn't it, Nandini? This strand is not coding for anything. You are calling it as coding strand. Why? Because it is having same information just like the RNA. The only difference is at the place of thymine, uracil will be there. Okay, at the place of thymine, uracil will be there. One more thing. But sometimes in the paper they ask you where that promoter and terminator is present. Is it present upstream of structural gene or the downstream of structural gene? Or they can also ask you it in this way, that promoter is present at the 3' end or at the 5' end. Yeah, simply promoter is present at the upstream of the template strand or the downstream of the template strand, like this. You have to remember one thing. Whenever we are using the terms upstream or the downstream, it is nowhere related to, big and small. It is like that. This side upstream, this side downstream. That's all. That's all. So if there is a question that at which end promoter is present, at which end of the coding strand promoter is present, like they ask now at 5' end promoter is present or at the 3' end. So remember, they will give you the reference. If they are taking the reference of template strand, in the question it will be clearly specified, right? Mostly we take the reference of coding strand because coding strand is a crybaby. Coding strand is like you are not you're not copying me. At least take me as a reference. Coding strand is like this. You're not copying me, but please take me as a reference. Okay, so if you are taking the reference of coding strand, then what will you say? That promoter is present at its 5' end, terminator is present at its 3' end. Okay, 3' end. If you are taking the reference of template strand, promoter is present at 3' end, terminator is present at 5' end. Keep it in your mind. Keep it in your mind. That's something important. It's simple as that. Like, see, if I'm facing camera like this, you're asking me, ma'am, where is the door? I will say right side, my right side. Or where is Almira? Left side. Now, if I'm like this, if I'm not facing the camera, okay, if you are asking me, where is Almira? I'll say towards the right side, and the door is towards the left side, like this, like this. So if you're taking the reference of template strand, your promoter is at 3' end. If you're taking the reference of template strand, your terminator is at 5' end. Here 5' end and the 3' end. So keep it in your mind, okay? Keep it in your mind. So see, so all the reference point while defining a transcription unit is made with the coding strand. So do not get confused if I'm using upstream, downstream words. So here in NCERT, they have clearly specified it, right? Please highlight this part. They have clearly specified it that all the reference point while defining a transcription unit is made with the coding strand. So to explain this, so here you guys can see they have given the sequence. Okay, so here you can see the promoter, the terminator, the start site as well. So promoter and terminator, they flank the structural gene in a transcription unit. Promoter is located towards 5' end, upstream of structural gene. And see, in the NCERT, it is clearly mentioned the reference is made with the respect of the polarity of coding strand. Okay, the reference is made with the polarity of the coding strand. That's why, because sometimes students are like, promoter is present at 5' end. Array, look at the complete question, read the complete question. If they are asking you that in context with the coding strand, where is promoter present, then it is 5' end. Otherwise, if it is template strand, then it will be 3' end. Is that clear? Sure. So it's a DNA sequence that provides binding site for RNA polymerase, and it is the presence of promoter in transcription unit, right, bache? So by switching its position with terminator, the definition of coding and the template strand could be reversed if we change the position of promoter and terminator. Fine, fine. So terminator is located towards the 3' end, downstream of the coding strand, and defines the end of the process of transcription. Okay, so as I said, there are certain extra regulatory sequences that may be present further upstream or downstream to the promoter. So some of property of these sequence shall be discussed while dealing with the regulation of gene expression. So yes, other regulatory genes are also there, like UTRs are also there, enhancers are also there. Okay, enhancers are also there. So we have covered this part about the monocystronic and polycystronic. After finishing this NCERT reading, I will tell you the importance of the split genes as well. Okay, we'll come to that point. Okay, so remember, a cistron is a segment of DNA coding for a polypeptide. Coding for a polypeptide. So if the structural gene, if it is monocystronic, like mostly in eukaryotes means it's not mandatory that they didn't mention only in eukaryotes, mostly in eukaryotes you are going to see. Okay, or polycystronic, mostly in bacteria or prokaryotes. So in eukaryotes, monocystronic structural genes, they have interrupted coding sequence. I told you they are split. Their coding sequence or the expressed sequence are exon. They are the one which will be there in mature or processed RNA. They are the one which will help to make the protein. Now it is making sense to you, yes or no? That's why I explained the topic first, okay? That's why I explained the topic first, so that you can understand the NCERT. Ultimately, bacha, it is the NCERT that you need to understand, okay? Ultimately, it is what? It is the NCERT that you need to understand. It is the NCERT you need to master. But to understand that NCERT, yes, you have to go little beyond NCERT in genetics as well, even in physiology, okay? So exons are interrupted by introns. So introns or intervening sequences do not appear in processed RNA. So this split gene arrangement complicates the definition of a gene in terms of DNA segment. That's a wonderful point. That's a wonderful point. So inheritance of a character is also affected by this promoter and regulatory sequence of structural gene, bache. So regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. Fine. And even bache, types of RNA and the process of transcription is also clear to you. I hope it is. Okay. So here they have simply mentioned that, see, the mRNA provides the template, tRNA brings amino acid, it reads the genetic code, RNA help in plays structural and catalytic role during translation. It plays catalytic role during translation. I hope you remember. So when RNA is acting like a catalyst, you will call it as ribozyme. So in translation, you talk about the peptidal transferase. In translation, you will talk about the peptidal transferase. We will discuss it. So actually that is a ribozyme. That's what you can remember directly. That peptidal transferase is nothing. That is what? That is a ribozyme. Okay. That is a ribozyme. Is that clear? Is that clear? So there is a single DNA dependent RNA polymerase that will catalyze the formation of all the RNA in bacteria, bache, right? So it will also use nucleoside triphosphates as substrate, the same way we have discussed it in DNA replication. So it polymerizes in a template dependent fashion, and you know that from the triphosphate, the energy that will be released, that will be used for polymerization. So it's ditto same like the DNA replication, right? The substrate, the dual purpose and all. Clear? Clear? So RNA polymerase will open up your DNA. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. So the RNA polymerase also. So this is the termination. So that's it. That's the process. Okay. Okay. So see, they are saying that RNA polymerase is only capable of catalyzing the process of elongation. Okay, so it needs the sigma factor to initiate the transcription. So sigma factor will be there. Once RNA polymerase start the transcription, sigma factor will say bye-bye to that RNA polymerase. That elongation part you can take care. I have just helped you in starting. Okay, so what is the role of sigma factor? Will carry that RNA polymerase to promoter. Once RNA polymerase get comfortable there, once RNA polymerase starts the transcription there, starts the formation of mRNA there, the sigma factor will say bye to that okay, done. So rho factor will help in the termination. Fine bache, fine bache. So in bacteria, mRNA doesn't require any processing to become active. So transcription and translation, they are taking place simultaneously there. Okay, both will be there in the cytosol, in the cytoplasm. Fine, both will be there in the cytosol, in the cytoplasm. But as I said in eukaryotes, there are two additional complexities. One is 3 RNA polymerases are there in nucleus, right? Right. So RNA polymerase 1 will form your rRNA. Here you guys can see the diagram as well. See the capping, right? And then the tailing, and this is the splicing. So spliceosomes are going to remove it. And finally, this is your functional mRNA. Finally, this is your functional mRNA. Clear, bache? Clear, bache? So see, these RNAs are formed by your rRNAs are formed by RNA polymerase 1, and RNA polymerase 3 will form tRNA and M5s RNA. Okay, okay, okay. And your small nuclear RNA. And RNA polymerase 2 will form your hnRNA. Here by mistake, I think, yes. See, even we need to revise few things. Sometimes even we make such mistakes. Okay. But here the wonderful part is, bache, here the wonderful part is that, you know, in the genetics, if you understand the concept once, na, then there is no need to, you know, revise that chapter entirely. If you know the concept, you just need to check few points. Like here, I made the mistake. I was telling you that your 5S will be formed by RNA polymerase 2 or snRNA. No, it is your RNA polymerase 3 that will form snRNA as well. That will form 5S RNA as well. That will form tRNA as well. RNA polymerase 2 will only form mRNA. Here I made a mistake, so please rectify it. Okay, please rectify it. Done. So the second complexity is that the primary transcript contain both exon as well as intron, and they are non-functional. So we have to go for the splicing. So introns will be removed, exons will be joined. So hnRNA will further capping and tailing. After capping and tailing, what will be there? You will get the mature RNA, that's all. Right? You are going to get the mature RNA, that's all. Clear, bache? That's all. Any doubt? Any doubt? So, see. Now, see the evolutionary perspective here. This is exon 1, this is exon 2, this is exon 3. You have to process this mRNA. You have to process this mRNA. You know, sometimes what we do, we only join exon 1 and 2. It's as per requirement. From this one mRNA, we can get different types of protein. Do you know that? From this one hnRNA, we can get different type of protein. If we are joining 1 and 2 exon, we will get different amino acid. If you are, we can also join 1 and 3 exon also. We can remove others. We can also just take second and third exon only. So if you are taking all the 3 exons, 1 and 2 and 3, accordingly you will get the protein. If you are taking 1 and 3, accordingly you will get a different protein. Only 1 and 2 different protein, 2 and 3 different protein. It's like that. It's like that. So from one hnRNA, you can process it in different ways as per the requirement, and accordingly you can get different mRNAs as well. So in eukaryotes, that's how it works. Clear? In eukaryotes, that's how it works. Is that clear? Yes, bacha? Is that clear? So tomorrow, right, it will be a marathon session, right? It's a kind of final marathon, like you will be discussing genetic code, translation, operand, HGP, Human Genome Project, and the mutations. So tomorrow, at any cost, we have to finish it. So I will start your lecture at 11, and hopefully till 3 p.m. or 4 p.m. I'll say bye-bye to you. Okay? Maximum till, chalo, I'll keep it till 3. Okay? I'll keep it till 3. Sure? Nandini, okay, then you, at that time you can leave the class. I'll try to, finish it before, but yes, we have to discuss it in detail. Done? So I'll share the PDF in official Telegram group. I will share all the PDFs together, as I said already. Okay, so that's all, bache. So today I'll discuss the syllabus changes as well. Fine, I'll discuss the syllabus changes as well. I'll keep a separate session to update you about the strategy about the syllabus changes. So don't worry about that. I'll take your half an hour. And if there will be queries, I'm going to answer that too. So now you know what you have to do. You have to put a wonderful comment in the comment section. You have to tell me you like the class or not. And if it is actually helpful, then please mention it in the comment section. Because you know that appreciation, even it drives us, okay, to do the better work. Fine. So thank you. See you all tomorrow at 11. Stay tuned to this channel. If you're new, do subscribe our channel. Fine, do subscribe our channel. So take care. Bye-bye. Thank you, everyone.